Well, we can hope. So I'm going to be giving you an overview of the Intellectual Disabilities Gene Function and Pathway Database, which is part of the IDDRC molecular core. I am responsible for this, as is Xiaolu Sturgeon, who is an MD PhD working in my lab, who's done most of the computer and bioinformatics analysis and setup for this database. And you've got the website address right here. Not the easiest address to remember or use, but that's what they gave us. And then there are email contacts for both myself and Xiaolu. So what are the goals of the database? The primary goal here is really to facilitate research on intellectual disability. And in order to do that, one needs to know what genes are involved and what those genes are doing, therefore the pathways in which they function. So what this database is designed to do is to consolidate information. So we are not making up new information. We're not generating new information. We're going out into the wider world and collecting information that is already there. And this includes, as shown here, genetic, clinical, metabolic, behavioral, therapeutic, and bioinformatics data. This is the goal of the database. So it is important to mention that this database is under construction. It is in no way in its finished state. So we want to provide the data, sort of a one-stop place to get information on a variety of factors. And as I said, all of this information is present in public databases, but researchers don't always have the expertise or the experience to access that data or even know which databases to go to. So the idea here is to assemble this information all in one place. And in addition to that, Xiaolu has been developing search tools which are designed to be user-friendly whether you are a basic clinical or translational researcher. So what is the content? As I said, this is information that comes from publicly accessible databases, and our database also provides links to these databases or to other databases to get additional information. So the current content of the database includes close to 300 genes. These have been retrieved from PubMed and OMIM, 300 genes that are known to cause intellectual disability when mutated. And this can be either primary intellectual disability or secondary to some other genetic defect. So in addition to this information on simply gene lists, associated with this is the disease, when these genes are mutated, which cause. We also provide gene function in what are known as GO terms or gene ontology terms. This is sort of a uniform standard for describing gene functions. We also provide basic information, gene name, aliases, so you don't need to know the correct name for your gene. If it is a bona fide alias used in the literature, you can use that. We also provide accession numbers. We provide information on where the gene is located in the human genome, which chromosome, down to the base pair nucleotide structure, or nucleotide, and we also provide information on the intron-exon structure and links to one of the publicly available genome browsers that contains basically the human and the mouse and various other genomes. We also have, at the moment for chromosome 21, but can implement this for all ID genes, information on gene variation. This includes SNPs that have been retrieved from dbSNP, but also includes the location of the SNP and the location within functional domains and information on whether this changes the coding region or what. And we also, in a separate place at the moment, provide information on protein interactions and networks. And you can generate your own networks depending on the genes or the proteins that you are interested in and user-specified queries. So this is the current home page for the ID Gene Function and Pathway Database project. Shown here in this blue bar are the features that we intend to have. This is protein-protein interaction data. Can't read that. Oh, homologs, so this would be model organism information for these same genes. We also have a tutorial that will eventually be posted here, additional links, and then publications. But what I'm going to show you today is a bit of usage on what is known as the ID Gene Quest. So what you see here is there are 3 choices of query types that one can use with this ID Gene Quest. It's very hard to find the arrow. So there are 3 types of searches that you can do. You can start with a gene name, you can start with a genomic region, or you can start with information on function and clinical disorder. But I want to show you 3 other things first. If you don't know what gene you want to search for, you can look at a list of ID genes. If you don't know what kind of function you might be interested in or what kind of clinical disorder, we provide lists of those. And some examples are shown here. So for ID genes, this is simply a list of genes and a description of their functions. These are ID genes that are currently in our database. Alternatively, you can look on a protein function. For example, we have hydrolases, we have receptors, we have kinases, and so on. So you can look for a type of gene function. If you're interested in kinases, I have an example that I'll show you. So you can look for all ID genes that are known to be kinases. And alternatively, you can look through a list of clinical disorders. So that is all information that you can do a simple search, or you could just scroll through and browse. So for the actual search page, I've got 2 examples that I'm going to show you. This is an example of a search for functional and clinical disorders. And here you have a choice between having it search through all human genes, or, as indicated here, you can search only for all ID genes. So you can narrow your search a bit. So in this particular example, we actually searched on all ID genes. This is a drop-down menu. And we searched for all ID genes that are classed as kinases, so have protein phosphorylation activity. And then you have options for filling out other features that are shown here. Anyway, so if you fill out simply query all ID genes that are kinases, what is shown in this yellow table here is the results list. And what you are provided with then is the gene name, an accession number, the RefSeq accession number, a direct link to OMIM if you want to go and look at the information that is there, a brief description of this gene, and then also a list of clinical disorders shown here. And so you can see some of these are what we would call probably metabolic diseases, glycerol kinase deficiency, but then down here we also have mental retardation that is X-linked. Here. So anyways, this is providing you a list of all kinases that have mutations which cause intellectual disability. Also included on this is strand, some very specific information on strand, the genomic start and stop, and the exon number in the gene. What we forgot to put on here is the chromosome, which we really need to add, and that's because we have this chromosome 21 focus. But chromosome number needs to be on there as well. So this gives you, and these are highlighted, this table is in yellow, because what we plan to do for everything in the database is every ID gene will be highlighted. So if you get a list of genes, you can immediately see which are our ID genes. All of these are highlighted because we searched on all ID genes that were kinases. So this is the slide that I included for Gunter. And I think this is a handy tool for what is becoming increasingly found in the clinic, I think, is a patient presents with intellectual disability, but there is no known chromosomal abnormality. It's not Fragile X. It's not Prader-Willi. It's not, they've ruled out the sort of standard things. It's not Down syndrome. And so what they are finding now is by screening these genomic tiling arrays is there may be copy number variation, deletion or duplication of genomic regions. And I think this is a handy tool. What we can do here is you simply select an alternative to what we looked at before, and that is the genomic region. You type in, so this is the drop-down menu where you get to choose. So you choose genomic region, and then here in this page, which comes up next, you type in the genomic location of interest to you. Whoops. Well, where the red arrow is, you simply type in the chromosome and the start-stop of the data that was obtained by doing one of these genomic arrays, for example, and you hit the Go button, and what is shown in this table, then, in this table here, is a list of all genes that are present in this genomic region, and in this column here, we have listed the disease phenotype if there is an ID gene in this region. And so in this particular case, I pulled this out of the literature from exactly this type of experiment, and there were 2 ID genes located in this region. These are not yet highlighted in yellow, but they are going to be. And in addition, you can see on the right-hand side of the slide the genomic organization of this entire region. And it's drawn to scale. And you see a list, red and blue indicates forward and reverse strand. So that's, I think, a handy tool for clinicians, for diagnosticians, and for researchers who are interested in furthering information on genes that can cause intellectual disability. So the last tool that I want to show you is just one example of the protein interaction tools that we have. This is not yet located here on site. This is a collaboration between Chris Cios, who's at Virginia Commonwealth University, used to be here, moved about a year ago, and my lab here. And this data at the moment is located at this particular website. And this allows you to ask for any gene, any protein you are interested in, you can ask, is there a connection to any other gene? So I have this gene, is there a connection, a protein interaction network between this gene and my ID gene? And also you can ask between this gene and any ID gene. Those are the query tools that can be done here. I will mention that Xiaolu has plans to upgrade this particular tool and bring it in-house. And the reason for that is that currently these tools run using Java, and Java can be very, very slow to load the data. So this data is not there. The analysis is done when you set up your query. For users who want to look for protein interactions, when you go to the protein interaction page, the first thing you will see are some instructions on how to set the Java run parameters on your own computer to get this to run reasonably fast. But you do have to be patient for loading the data. And so it's trivial to change those run parameters and things will do better. And so the example here is simply of asking for protein interaction paths. The user specifies 2 proteins, the start protein and the end protein. You can select from a drop-down menu. If you don't remember the actual gene name, you do need to have the correct name here. So you can look at the drop-down menus. You put in your start and stop protein, and this is an example of the results. And so you have, this was simply a query between the SOD gene on human chromosome 21 and the histone deacetylase 1 gene, whose chromosome location I don't know. Anyway, on the left-hand side of the black box, you have your start protein, and on the right-hand side, you have your end protein. And for this particular query, we chose a maximum of 5 steps. The user can specify this. I only want to look at direct interactions. I only want to look at interactions where there's one protein intervening, or so on. And what you get is a list of all paths between these 2 proteins, and you can mouse over, when it's live, the gene name, and you will get this kind of information on what the gene is. And what we will do here again then is to highlight all ID genes. And another example of this kind of thing is a query that was done between an X chromosome linked mental retardation gene shown here and neurofibromatosis 1, which also causes intellectual disability. And rather than highlighting at the moment, I have simply circled proteins that are also known to be involved in intellectual disability. I would say that this is a hypothesis generating tool. Okay, this gives you a different view of what your protein or gene function might be. Very user-friendly. I think all you need to know is your start and stop. So the very last thing is, at the moment, the SNP data is only set up for chromosome 21. If there is interest, Xiaolu could expand this until we can look at all ID genes or all genes in the human genome. And basically what you do with this is you choose the gene that you're interested in, you select the type of SNP that you're interested in. Non-synonymous and non-conservative are the amino acid substitutions are the SNPs that are shown in this query. Non-synonymous means there's an amino acid change. Non-conservative means they don't have the same biochemical function. And what Xiaolu has done here then is merged information on protein domain. So for every protein in here, she went to the SMART database and annotated every region of the protein that has, with its functional association, a functional domain, a kinase domain, a transmembrane domain, so on, and coupled this with SNP data from the SNP database at NCBI. And then what you get here then is a list of all SNPs in your query gene, and it will tell you where it is in the protein, and if it is in a functional domain, it will tell you what that functional domain is. Why are you interested in this? This may or may not lead to intellectual disability. It is another way of looking at natural variation, natural nucleotide polymorphisms, and can you predict that there might be any functional implications? So, how do you use this database? It is freely accessible. Just go to the website address. At the moment, don't try this at home because we haven't yet got access off campus. Kathy Acha is working with us to get that done. And I emphasize again that this is under construction. The things that we would like in particular, I think, to add would be model organism data. There are people here who work with zebrafish, who work with mouse, who work with yeast, whatever. Is an ID gene in human? Is there an ortholog in one of these organisms? Or is there a gene that someone is interested in zebrafish that is an ID gene in human? We are going to upgrade, as I mentioned, the protein interaction data for speed and efficiency. We're going to add more protein complex information from mass spec experiments, and we're going to add external accessibility, which means basically anyone in the world can access this. So I've shown again the two website addresses. If you contact me, if you want to talk about how to use these tools, if you have problems with it, if there are new tools you would like to add, if you see errors or genes, we know we don't have all the ID genes. So if you want to tell us, give us the RefSeq for ID genes, we will add these. If you've got complaints, you don't like this, let me know. If you have problems with accessibility or running some of these tools, if there seems to be an error in that, please contact Xiaolu. And so that's all I have to say. Be happy to take any questions from anybody here.